Well, hello and welcome back to Science in the Age of COVID-19. Today we are extremely excited to welcome Paul Bieniasz from Howard Hughes Medical Institute and the Rockefeller. Paul's lab has a great track record in studying the genetic warfare that constantly goes on between viruses and their eukaryotic hosts. Prior to the COVID pandemic, his lab specialized in various retroviruses such as HIV and related primate immunodeficiency viruses. His lab has also greatly expanded the toolkit for what is known as paleovirology, looking at the signatures that retroviruses leave behind in their host to decipher what sort of battles were taking place and who eventually won. Like many of the other speakers in our series, Paul's lab, once the magnitude of SARS-CoV-2 became apparent, he quickly turned the lab around to focus on this. They helped develop and characterize various serological assays, and they are focusing most of their effort on development and characterization of therapeutic antibodies and studying how viral strains can escape from them. So Paul will give us an update on their progress. Okay, Paul.
Okay, thank you, Lauren, for that very kind introduction. And thank you and Sarada for the invitation to speak to you today. As Lauren mentioned, until earlier this year, I was primarily a retrovirologist studying the sort of interaction between retroviruses and their hosts. But when SARS-CoV-2 came to town, we pivoted, and it became somewhat obvious to us, and I guess to many people, that one of the main ways that we were going to find our way through this pandemic was through the activity of neutralizing antibodies. That would be true whether that was through acquisition of immunity via natural infection as a response in terms of vaccines, through the use of convalescent plasma therapy, or potent monoclonal antibodies as therapeutics or prophylactics. And I'll touch on each of those aspects as I go through the talk. So in about early March of this year, we really set ourselves up to develop robust high throughput assays to try and measure and characterize neutralizing antibodies to SARS-CoV-2. So the target of neutralizing antibodies on SARS-CoV-2 is the spike protein. It's this protein that decorates the exterior of viral particles. It's somewhat typical of the type of fusion machinery that you would find on many envelope viruses. It makes a trimer with a receptor binding outer domain, and then a transmembrane domain that really has the machinery that drives fusion. It is slightly unusual in that it's very large compared to most viral fusion proteins, but in many other respects, conceptually very similar. It has a receptor binding domain that'll be pivotal to what I talk about, that adopts one of a couple of different conformations, folded flat against the spike or in a sort of a projecting conformation that allows it to interact with a receptor called ACE2. So in order to develop assays to conveniently and rapidly measure the activity of neutralizing antibodies, we've developed a number of recombinant viruses, either based on HIV-1 or VSV, where we've replaced the outer envelope of those viruses with the SARS-CoV-2 spike proteins. Each of these viruses is armed with a reporter gene, either GFP or nanoluciferase, so we can simply measure infection as an expression of that reporter gene. And so we've also made cell lines that express the virus receptor, ACE2, and we can just mix these things in a microtiter plate and do very simple GFP or luciferase assays to measure infection. And the blockade of that infection using neutralizing antibodies. So here's one of our workhorse assays. It uses a replication defective HIV-1 that carries a nanoluciferase gene. And so if we infect cells with this virus, we get lots of luciferase activity in those cells that's not blocked by normal plasma. But if you add increasing amounts of plasma, depending on how potent that plasma is from a COVID-19 convalescent, you can inhibit that infection. And we designate the potency of that plasma as basically the dilution that can inhibit about half of the infection. Okay, so these are all fake, not real SARS-CoV-2 viruses, but for each of them, what we've done is to make sure that they represent the neutralization properties of the authentic SARS-CoV-2 virus. So here's that 50% neutralizing titer for SARS-CoV-2 itself, and then one of our surrogate viruses. And you can see the correlation isn't perfect, but it's pretty good. And we can do these assays at a rather high throughput to measure neutralizing antibody activity in a variety of contexts. So here is a somewhat important piece of data. Well, this represents 450 or so convalescent individuals arranged from left to right on the x-axis in increasing order of how potently their plasma neutralizes infection by one of those pseudotype viruses, as shown here on the y-axis. And what I want you to notice is the rather extreme variation in the potency with which plasma neutralizes SARS-CoV-2 infection. There are a significant number of individuals, around 20% or so in our particular assay, that have very low levels of neutralizing antibodies, less than 1 in 50 neutralizing titer 50. At the other end, there are about 10%, depending on where you draw the cutoff, of what we might call elite neutralizers, people whose plasma you can dilute thousands of fold and it still neutralizes the virus. But what's sort of important is there's a very strong skew towards the bottom end of this distribution. This is in no way a normal distribution. There's a lot of people around with rather low levels of neutralizing antibodies. So what factors might affect where an individual might fall on this distribution? Well, we don't know all the variation, all the contributions, but we do know, for example, that males tend to have a little bit higher titer neutralizing antibodies than females. That's probably a reflection of the increased severity of disease on average in male populations. So if you compare people who've had severe COVID-19, people who have been on ventilators, for example, as opposed to those that have had mild infection, again, there's a very clear difference. And we don't know, but we strongly suspect that this is a reflection of the amount of systemic antigen to which each individual's immune system has been exposed. Basically more antigen, stronger immune response, higher titer neutralizing antibodies. As I say, we don't know that for sure, but it's a theme that I will touch on as we go through. So how long do those neutralizing antibodies last? This has been a subject of some controversy, and this is essentially what we find. So again, these two charts on the left, these are convalescent individuals, again, arranged from left to right in ascending order of plasma neutralizing titer. At the first visit, that's sort of about 1 to 3 months after they were diagnosed PCR positive. And then those patients came back for a second visit or a third visit just a couple or 4 weeks later. And you can see, even in that short time frame, that the neutralizing titers are declining. This is a cohort in the UK. This is a cohort that visited Rockefeller Hospital, where they've just been back for their 6-month visit. This is the distribution of neutralizing titers when they visited 6 months ago. And this is their more recent neutralizing titers. So you can see there is a clear and fairly dramatic diminution in neutralizing titers. That's represented in a slightly different way here. But basically, after about 6 months, it's sort of about 10 to 20% of the level of neutralizing activity that was already not particularly impressive at the outset. That decline is significant over over 6 months. And as I'm sure you've heard, there are reports in the press of individuals becoming reinfected, and depending on how the vaccine is rolled out, that's something you can expect, I think, to hear more of. This is in some ways sort of typical of experience with the coronaviruses, the seasonal coronaviruses that are known to colonize people and have done for many hundreds of years. So one of the activities we've been engaged in is to try and help clinical investigators understand what the results of serological assays mean. You can, I'm sure, appreciate that when someone goes for an antibody test that measures whether they have antibodies to SARS-CoV-2, that can be used to sort of diagnose whether someone's been infected, but perhaps just as importantly, in some ways, one would like it to be a prediction of whether that person will be immune to infection in the future, either having been infected in the past or received a vaccine. These type of assays also been used rather, and this is sort of a new use, to try and triage individuals who are going to donate plasma for convalescent plasma therapy. So we'd sort of like to know what each of those serological assays mean. And so here are two such serological assays that are developed using elements of the spike protein from these two manufacturers. And you can see that over time, they sort of maintain the levels of antibody, at least measured by this assay. These assays. However, if we take those very same plasmas and measure neutralizing titers, even though the binding antibody titers are maintained, neutralizing antibody titers are declining. So that's a sort of a word of caution in terms of using these assays to predict immunity and triage patients for use for donating convalescent plasma. So we've tried to correlate neutralizing titer versus titers on various assays using a number of assay platforms. And some of them do actually a pretty good job of predicting neutralizing titer. Here's the titers for neutralization and for the binding antibodies. Others do much less well, as you might expect if you're measuring antibodies to the N protein, a protein on the interior of the virus. That may not give a good prediction of neutralizing titer. The fact that it does give some level of predictive value, again, suggests to us that because these variables can't be causally related, there must be a third variable, and we suspect that that third variable is, as I've mentioned, just the level of systemic antigen exposure, really the amount of virus that replicated in that individual. I just note this assay on the top right here. This is the one that the FDA has picked out as being usable to discriminate which plasma units can be used for convalescent plasma therapy. And this is the titer that the FDA has designated as high titer above the line, low titer below the line. So anybody who gets plasma therapy under this emergency use authorization would have one of these units. My opinion is that that actually isn't a very good discriminating assay. I'd much rather have those convalescent units measured using this assay and say take the top 10% here rather than the top half here. Okay, so picking out convalescent plasma units for therapy is one use of such assay. Simply doing serosurveys, diagnosing who has been infected, is another use. And here we've also found that the commercial assays are somewhat variable in their performance. So some of them do quite well. So here's a time after PCR diagnosis for this cohort of sort of mild to moderate infected individuals who clearly they all had symptoms. There were no asymptomatics here, but were not hospitalized. If you follow those over time, most of the assays end up being quite good at diagnosing who has been infected, but this one particular assay from Abbott is doing really not very well after about 3 months. And now at 6 months, it's only really detecting about half of the people who have been infected. So that's sort of a public service announcement. If you go for an antibody test, make sure you know which particular assay is being used to measure whether you have actually been exposed to this virus. Okay. One other aspect of serological immunity that we have investigated and has actually been quite controversial is whether cross-reactive antibodies from the seasonal coronaviruses have activity against SARS-CoV-2. So what we have done is to take a bunch of sera from 2019, so pre-pandemic, individuals who were actually diagnosed by PCR with coronavirus infection with one of these seasonal coronaviruses. We've measured the ability of those sera to neutralize this seasonal coronavirus, OC43, which of the seasonal viruses is about the most closely related to SARS-CoV-2. And many of these plasma are very, very potent at neutralizing this seasonal coronavirus with titers in the thousands or so. None of those sera touch SARS-CoV-2 in this neutralization assay, different to COVID-19 sera. So our view is that there really isn't, at least at the level of neutralization, any particular immunity conferred by prior exposure to a seasonal coronavirus. There's another piece of data that I'll mention in a minute that is also consistent with that view. So we are really quite immunologically naive, at least serologically, to SARS-CoV-2. Okay, so let me move on now to talk a little bit about what we've done here at Rockefeller, a group of us that really started at the beginning of the pandemic. Michelle Nussensweig, Charlie Rice, our groups and my group really got together to try and identify potent monoclonal antibodies and bring them to the clinic, basically by using our sort of combined expertise. So this is essentially what we did. The procedure begins in Michelle Nussensweig's lab. What his group did was to take the receptor binding domain, as we knew that this was a target for neutralizing antibodies, and take white lymphocytes from convalescent individuals through a sorting procedure whose final step is whether those isolated B cells will bind to the receptor binding domain. Okay, so it's labeled with two different fluorophores, and these cells that bound to both colors of the RBD were then sorted. Individual cells were put in individual wells of a 96-well plate. Michelle's group then clone the antibody genes from each of those individual cells, express the antibodies, which then come to our lab, and then we screen them with one of our high throughput assays for the ability of these monoclonal antibodies to neutralize one of our pseudovirus infections. So we've now done this for hundreds of individual human monoclonal antibodies. Here's a sample of the data, and highlighted in the colors are some of the antibodies that we've been particularly interested in. Some of them don't neutralize at all, and some of them are really, really quite potent. And I'll go into that over the next few slides. Just a word or two about what Michel and his group found when they were doing this antibody sorting. So these are 6 individuals, and the numbers in the middle of these pies represent the number of antibody sequences that were obtained from those individual B cells. And the slices of these pies that are colored or shaded in here represent expanded clones. Individual B cells, or what I should say, the daughters of individual B cells that were found multiple times during this sorting procedure. So they represent cells that have encountered antigen and then have proliferated, and the rearranged antibody genes you find on multiple occasions. What's interesting is that you don't just find the same clone in single individuals, you find more or less the same antibodies represented by colors here in different individuals. So this red antibody was found in these 2 people, this orange antibody here and here, the yellow antibody here and here, and so on. So these represent nearly identical expanded clones in different individuals. What's also interesting about these antibodies is they have quite a low degree of somatic hypermutation. They represent what looks like a de novo response. They're not a recall response from a prior infection. And even though these cells are making antibodies that bind tightly, as we'll describe, to the SARS-CoV-2 RBD, the germline sequences of those antibodies seems already very good at recognizing the spike protein without large degrees of somatic hypermutation. So here's another somewhat important piece of data. If we take these individual monoclonal antibodies and ask how potent they are at neutralizing SARS-CoV-2 virus, we see a range of potencies. Some are not very potent at all, but some of them are extremely potent, okay? With IC50s in the low nanogram range. What's interesting is that the potency of these antibodies doesn't seem to bear really any relationship to the potency of the plasma from which that individual was obtained. So this individual, for example, very low titer plasma, lots of potent neutralizing antibodies, whereas this person, very, very potently neutralizing plasma, not really any more impressive in terms of the potency of the monoclonal antibodies. What this tends to suggest is that pretty much everybody is capable of making these potent neutralizing antibodies. And really the difference between individuals is just how much their B cells have expanded and how much of those potent neutralizing antibodies have been made into the plasma to give these neutralizing titers. So equally potent antibodies, just different amounts of them in different individuals. And some of the antibodies that we're going to move forward with in terms of the potent monoclonals, some were cloned from people who really had unimpressive neutralizing titers at this end of the curve, others from individuals who had plasma that was very much more potently neutralizing. So we've moved forward with 3 antibodies. That's sort of been funneled down to 2 antibodies. Here are their neutralization curves against the SARS-CoV-2 pseudovirus, one of those fake viruses that we've been using for screening. And then here against the authentic SARS-CoV-2 virus. So as I mentioned, IC50 is in the low nanogram range, okay? These are sort of picomolar active agents, very potent antiviral agents. So in collaboration with Tim Sheehan and Andrea Schaefer at UNC, we've taken these antibodies into sort of animal model experiments. Actually, the antibodies have gone into a number of animal models. I'm just going to tell you about this particular one. This employs a mouse-adapted SARS coronavirus 2. The mouse adaptation really is 2 amino acid substitutions in the receptor binding domain that allows the virus to use the mouse version of ACE2. Okay, so the wild type spike protein works very nicely on human ACE2 here, but doesn't work very well on doesn't work at all, in fact, on the mouse ACE2. But you make these 2 amino acid substitutions to generate this virus SMA, and now the spike protein is able to infect cells with human ACE2 or mouse ACE2, and in fact can be used to infect mice. So because we're messing around with the RBD, we have to understand what effect that has on antibody sensitivity. And the 3 antibodies that we're particularly interested in, their potency against this variant virus is just the same as it is against the wild type virus. There's the odd antibody that loses the ability to neutralize the mouse adapted virus, but we're not taking those forward. So here's an in vivo experiment where mice were given one of the 3 favorite antibodies, okay, or a control, and then infected with this mouse-adapted SARS-CoV-2. And this is lung titers, plaque-forming units per lobe. And you can see the untreated animals, there's about 1 million infectious virus units per lobe of lung in those mice. But if they're pre-treated with a moderate dose of each one of these antibodies, then you get about 1,000-fold reduction in the amount of virus in the lung. Now, if you start combining the antibodies, and you'll see in a few minutes why these combinations were chosen, you can get even more impressive results. So again, very modest levels of antibodies. This combination in particular, you go from 1 million infectious units per lung down to almost sterilizing protection. Even this very low dose of antibody can reduce lung titers by about 100-fold. So these are the antibodies that are going forward in clinical development, and hopefully will be in clinical trials soon. So, of course, when one thinks about using antibodies, or really any agent as an antiviral, one of the first questions one has to ask is about resistance. Okay, how likely is it that resistance to these agents will emerge in SARS-CoV-2? And in thinking about how resistance might emerge, it's actually somewhat of a complicated question. So if you imagine a transmission chain of individuals where you start with an antibody susceptible virus, and then in this particular individual, an antibody resistance mutation emerges, and that virus is then transmitted. So you have an antibody resistant variant. The probability of such an event occurring is affected by a number of different variables. So the replication fidelity of the virus itself, its propensity to generate mutations, which we know is low for SARS-CoV-2. It has a proofreading polymerase and actually makes mutations at a rate that's significantly lower than most RNA viruses. But of course, the antidote to replication fidelity in terms of exploring genetic diversity is population size. So if you have large populations, and we know, of course, that the global population of SARS-CoV-2 is enormous, then basically it's inescapable that every possible single nucleotide mutation of SARS-CoV-2 that could be generated has at some point been generated. And if you do the maths, it's quite likely that a substantial fraction of even of individuals harbor mutations covering a large fraction of the possible mutations that SARS-CoV-2 could explore. So, but on top of those sort of virus intrinsic properties, things like transmission dynamics can affect how likely it is that antibody resistance will emerge. If you look at this graph, this chart on the lower left, which I made up, basically shows a peak and a diminution of viremia in an individual, and with it, presumably the probability of transmission. The degree to which that trajectory overlaps with the generation of neutralizing antibodies will affect the frequency with which virus encounters antibody, and therefore the selective pressure that's placed on that virus, and in turn, how likely it is that such an antibody resistant virus would essentially rise to the surface of a viral population and be transmitted. Okay. Those parameters are obviously affected by things like waning immunity and reinfection, how the monoclonal antibodies are deployed, convalescent plasma therapy, and of course vaccines. And convalescent plasma therapy, I'll just highlight, this has been given to tens of thousands of individuals, often without any knowledge of the titer of the antibodies in those convalescent plasma units. So it's sort of inescapable that many viruses have been exposed to sub-therapeutic levels of neutralizing antibodies. Bearing in mind, we're cloning these antibodies from convalescent individuals. So we're really talking about, at least to some degree, overlapping antibodies. And so how this all plays out in terms of how likely antibody resistance will emerge to SARS-CoV-2 is really, I think, somewhat unclear. First time there's been a global pandemic where we're going to be deploying monoclonal antibodies, convalescent plasma therapy, vaccines, and likely that reinfection will occur. And so it's, I think, quite important to try and understand how antibody resistance might emerge. So as I say, we don't really know, but we have started to develop tools that can help us get at that question. And one of the tools is, in fact, a chimeric virus that I mentioned at the beginning. It's a recombinant vesicular stomatitis virus where the envelope protein has been replaced with the spike protein of SARS-CoV-2. So it replicates as VSV, but uses ACE2 as a receptor and has the neutralization properties of SARS-CoV-2. We do this because VSV doesn't have a proof-reading polymerase. So with small laboratory-generated populations, you can explore a much wider degree of genetic diversity. So when we first built this virus, it actually didn't replicate that well. We had to adapt it in cell culture. In so doing, it picked up a couple of mutations. These are the ones we think are functionally important. These are probably passenger mutations. So we're using 2 different virus strains for all our subsequent experiments with this virus to account for that. But this virus grows extremely robustly. We can generate plaques in 1 to 2 days, and it grows to titers of 10 to the 7, 10 to the 8. We can very easily generate the required diversity for diverse viral populations to test for resistance to antibodies. Very, very importantly, of all the viruses that we have made, the fake viruses we've made, this virus most closely mimics the neutralizing properties of SARS-CoV-2. So here are the IC50s for 20 monoclonal antibodies against SARS-CoV-2 on the y-axis and this recombinant virus on the x-axis. So you can see that it actually very nicely recapitulates the neutralization sensitivity of SARS-CoV-2 itself. And we have a couple of versions, one with GFP that you just saw and another with a luciferase reporter for sort of more easily quantifiable results. So this is basically how an experiment that explores antibody diversity works. So we take a large population of about 1 million members of this recombinant virus that's been passaged for a little while to generate diversity, carries a green fluorescent protein reporter. We mix it with very high quantities of monoclonal antibody, way higher than the IC50, put it on cells, and then any viral variants that grow out, we passage, again in the presence of antibody, and then start sequencing, isolating individual members of a putatively resistant population. I'll come to this part of the experiment in a few minutes. So this is what such an experiment looks like. This is 1 well/12 well plate where we've infected cells with 1 million infectious units of a GFP reporter virus. And if we do that in the presence of 10 micrograms per mil of one of our favorite antibodies, we kill nearly all of the infection. Nearly, but not all infection events. So if you look at this particular part of the cell monolayer, you'll see that some cells have been infected and it looks like we're forming a focus of infection here. Same down here. If we zoom in on this part of the plate, you can see a little group of cells where it looks like one virus has infected the monolayer and then has started to spread. So that's indeed what happens. So if you take supernatant from these cultures, put it onto fresh cells in the presence of antibody, in a couple of days, you get the same picture here. So then if we start sequencing what has happened, this is what's happened. So what I'm going to show you now is a few charts where the sequence of the spike protein is represented as a codon position on the x-axis, and then the frequency with which we find variants at each codon position is represented on the y-axis. So if we grow the virus in the absence of antibody, there's very little variation, low frequencies of variation. But if we do this in the presence of this particular antibodies, you can see at a particular position, you start to see high frequencies of variance. Similarly with this antibody, different positions are selected, but there's high frequency variation, and so on with this antibody. Okay, if we isolate those individual variants by plaque purification and then start to sequence and characterize them, this is what we find, okay? So the wild type virus is potently neutralized by all 3 of our antibodies, but viruses that have been grown in the presence of these 2 monoclonal antibodies, you start to see variants. So here's one such variant, Q493R. It's now completely resistant to this antibody and this antibody, but retains complete sensitivity to this antibody. Conversely, we grow virus in the presence of this antibody, C135. We get this mutant, completely resistant to that antibody, but retains complete sensitivity to those two antibodies. So this begins to tell us where the antibodies are generating resistance. Then we went to the sequence database and found that sometimes those variants occur naturally. And so what we've done is to explore the naturally occurring variants in the RBD to see if any more of them confer resistance to these antibodies. And so what these three charts show are lists of mutants, naturally occurring mutants in the SARS-CoV-2 RBD that are circulating in global populations. And on the y-axis, the IC50s for each of our favorite antibodies. And you can see that C121, this set of variants here confer nearly complete resistance to this antibody. There's a significant overlap in the mutations that cause resistance to C144, whereas it's a completely different set of variants that cause resistance to C135. And you can sort of use that to map the neutralizing epitopes of these antibodies. And of course, that gives you a lot of information about how you might want to combine these antibodies as therapeutic cocktails. But before we do that, let's just look at the global sequence diversity across the spike protein. So again, we've got the spike protein codons represented on the x-axis, how frequently variants are represented at each position on the y-axis. And each one of these symbols represents an individual mutation in the sequence database. And the colored triangles here represent mutations at positions where we know that that would confer a resistance to one or more of the monoclonal antibodies. And so in red and green are the resistance mutations that we found for the antibodies that we developed at Rockefeller. And in blue, also in red, because there's overlap between our antibodies and the Regeneron antibodies, the blue and the red show you positions where mutations that confer resistance to the Regeneron cocktail are found. So we shouldn't, I think, pay too much significance to what the actual position on the y-axis is at this point, because there's quite a lot of sampling bias. If a particular sequencing center happens to be close to a set of viruses where there's a founder effect and a particular variant is found in that locale, that can distort to some extent, to a large extent, the actual measured frequency of each variant in the global population. But what I would point out is that each one of these resistant variants, they're all present at 1% or less of the globally circulating populations. And they're present at a frequency that's sort of unexceptional compared to all the variants at other positions. So at this point, there's not really strong evidence for selection at these positions. There's also not really, selection either in a positive sense or a negative sense. They're not being purged from the viral population, but they don't, at this point at least, seem to be rising to the surface. Okay. Nevertheless, data such as that does inspire us to combine antibodies and cocktails. And so just to show you that that works very nicely, here's the same experiment that I showed you a minute ago, but in this time, instead of using 10 micrograms of a single antibody, we've sort of split the dose between two different antibodies that we've chosen to have non-overlapping resistance profiles. And you can, as you can see, while we still get individual cells getting infected because we haven't got enough antibody there to neutralize every last one of these million viruses, when those viruses do infect the monolayer, they do not appear to spread. And if we try to passage virus in the presence of antibodies, when we're using one antibody, we have just as much virus there as if there was no antibody, even after the second passage, because the resistance occurs that quickly. But if we have combinations of two antibodies, then we basically crush replication, and really there's no virus present after two passages. Okay, so nevertheless, we still have to bear in mind that the monoclonal antibodies that we are working with, they are derived from a set of antibodies that are circulating globally, that are generated in response to natural infection and potentially in response to vaccines. So to what extent is there going to be overlap in the resistance profiles of monoclonal antibodies and polyclonal sera? To what extent are sera that are generated following vaccination or natural infection, are they polyclonal and variable between individuals? Okay, that's sort of an important question in terms of thinking about what the impact of resistance to individual antibodies is going to be in the broader context. So to try and get at that question, we've done basically the same experiment, but this time instead of using a monoclonal antibody, we're using a polyclonal plasma, convalescent plasma from an individual. Sometimes we need to passage the virus to a larger extent to generate those resistant viruses. So what do we find? So actually, it turns out things are quite variable when we do this experiment with plasma. For some plasmas, it looks like those convalescent plasmas behave a little bit like they're almost solutions of monoclonal antibodies. So this individual, for example, even after the second passage, we selected high frequency variants at a single position. For some other plasmas, it's really, really hard to generate resistant viruses. And then for this individual, we failed completely. Even after 4 passages, there's really no change in the sequence of the virus that's been passaged in enough plasma to suppress replication, but not kill it completely. Other plasmas have an intermediate phenotype. We can see variants beginning to arise at passage 2, while at passage 4, those variants are increasing in their frequency. So quite a bit of variation between plasmas. Now, we've done this experiment now with about 25 different plasmas that have good neutralizing activities. And this represents a summary of all of that data basically combined into one graph. So each point here is a variant that has appeared when we've grown that virus in the presence of one or other plasmas from convalescent individuals. And so what we think we're beginning to see is the designation of what we think of as neutralizing epitopes, areas on the spike protein where plasma is exerting selection pressure. So we haven't yet characterized all of these individual variants. I'll show you an example just of 2 where we have those. So here are mutants that were selected by growing the virus in the presence of plasma from this individual, COV47, and it selects for mutations at around amino acid 150 in the spike protein. And we can see that those mutants are about 10-fold, 8 to 10-fold resistant compared to wild-type virus to plasma from that individual. But they have significantly lower degrees of resistance, if any at all, to plasma from different individuals, okay, suggesting that these plasmas, their neutralizing activity is dominated by different antibodies. Similar situation here, mutant selected using this plasma, COV-NY, selects for variation at this position here around amino acid 444, 445, and those mutants, they have about 50-fold resistance to that plasma from that individual. But those mutants are not resistant to plasma from different individuals. So what that tells us is there does seem to be a significant degree of polyclonality and difference between individuals in terms of the antibodies that are dominating their plasma in terms of neutralizing activity. That's quite good news if you in terms of thinking about how likely it is for viruses that are broadly resistant to neutralizing antibodies to arise. So this sort of summarizes everything we now know about resistance to antibodies. So what's shown here are pictures of RBDs where we or others have generated resistance to monoclonal antibodies. These are the Rockefeller antibodies or the Regeneron antibodies. And these are RBD mutations where we found resistance mutations in plasma. And you can see there is some overlap between them. Okay, so some of the plasma antibodies are, of course, they're giving antibody specificities that overlap with those from human monoclonal antibodies. More broadly, in the spike protein, everything that's colored in red here is a variant that appears to have been selected by replication in one or other plasma. But I guess the take-home message here is that there is a significant degree of variation between individuals and even within an individual in terms of where on the spike protein neutralizing antibody would bind. Okay, so just for the last minute or so, what I want to tell you about is some very recent data that we've found. This is having individuals come back to Rockefeller 6 months after we initially cloned antibodies for them. And as I told you earlier, those antibodies had very low levels of somatic mutation. Okay, so these are number of mutations in the heavy chain and light chain of antibodies about a month after they were infected. 6 months later, the number of mutations has clearly increased. The antibodies clearly are evolving, or have evolved over that period of 6 months. And some of them have evolved in quite interesting ways. So here is an antibody, C144. It's one of our favorite antibodies that's going forward in clinical development. And I've already told you about resistance mutations that can be selected with that antibody. And we find that those mutations that we can generate in cell culture and also exist naturally confer complete resistance to this antibody. Now, what has happened in this individual is that this antibody has evolved, and there are several derivative variants. Here's one of them. So it's the same antibody, just with more somatic mutations. That antibody has clearly evolved and now can actually neutralize those previously resistant variants. So it's clear that over time, the antibody response is maturing and perhaps broadening to include virus variants that otherwise might resist that initially generated close to germline antibody. And some very remarkable data from a collaborator of ours, Saurabh Mahandru, might give some clues as to how this is happening. What he's found by taking gut biopsies from individuals as long as 3 months after symptom onset. These are people who have recovered. They have no virus in their nasal swabs. But if he takes gut biopsies and stains for SARS-CoV-2 antigen, he can find cells. This is in the duodenum of these individual cells that appear to be positive for virus. They're not present in the pre-illness gut biopsies. So this suggests that there's a degree of persistence in SARS-CoV-2 replication, and he finds this in about 1/3 of the 15 or so individuals that he's looked at. Persistence of virus that might explain this maturation of the antibody response. Okay, so let me summarize what I've told you. So convalescent SARS-CoV-2 neutralizing antibody titers can vary very dramatically between individuals. Some of the serological assays that are employed clinically are actually quite good at predicting those neutralizing titers, at least in early convalescence. The neutralization titers, though, decline, and during the first 3 months, they have a half-life of about a month, a little over a month, and by 6 months, they have declined around five-fold or so. We can clone neutralizing antibodies from convalescents that are quite good as antivirals. They protect in in animal models. There are antibody escape variants in natural populations. At present, they're at very low frequencies. So something to keep an eye on rather than to get really panicked about. And the neutralizing antibody response to SARS-CoV-2 is polyclonal. It's quite variable within and between individuals in terms of its specificity. And as I just showed you, it seems to continue to evolve over months in at least some individuals. Okay, so let me wrap up quickly just by acknowledging the people that contributed to this work. And what I would say is that when this virus came to town, we really started doing science in quite a different way. So everything I've told you has been sort of the contribution of a much larger group than just my lab. We've been working very closly with Michelle Nussensweig and Charlie Rice's lab on all of these projects, particularly in terms of developing those monoclonal antibodies. We've also worked quite closely with Pamela Bjorkman at Caltech. At UNC, Alexandra Schaefer and Tim Sheehan did the mouse experiment that I told you about. And just recently, we have started working with Saurabh Mahandru, who's made this rather remarkable observation that that virus seems to be persisting in the gut perhaps longer than we had previously appreciated. Some of our clinical work on the clinical samples has been done in collaboration with New York Blood Center, particularly Larry Lussinger. And I'd also want to acknowledge Helen Wise and Sarah Jenks at the Royal Edinburgh Infirmary in Scotland, who really helped us in terms of the longitudinal analysis and those pre-pandemic analysis of the seasonal coronaviruses. In my own laboratory, so Theodora Hadzioannou is my scientific partner, also happens to be my wife. We do pretty much everything together, so she's very much an equal contributor to what I've talked to you about. But I really want to highlight these individuals, Frauke Mutsch, Fabian Schmidt, and Jiska Weisblum. So when New York locked down, beginning in March, these guys went to work close to 24 hours a day, really working incredibly long hours, incredibly hard to get these projects off the ground and brought to some semblance of completion. Really a remarkable level of commitment. I'd also want to highlight these individuals who've also, in my lab, who've also contributed: Magda, Fengwen, Daniel, and Justin. And with that, I will conclude, and thank you very much for listening.
Oh, thank you, Paul. It was a very insightful talk on various aspects of antibody response to SARS-CoV-2. And of course, we have a lot of questions, so let's get right onto them. So I'm probably going to start off asking a question that's, I guess, in the mind of all of us out here. So given that we woke up to some very good news yesterday with the vaccine trials from Pfizer vaccines look pretty promising. So we are curious as to how long will the protection from neutralizing antibodies last?
Okay, so at this point we have to say that is unknown. I think a very, very important point to make, though, is the sort of apparent lack of durability that one might see in response to a mild SARS-CoV-2 infection. That in no way predicts the durability of an immune response to a vaccine. Okay, what we have seen in terms of the publications in terms of antibody responses to vaccines is they seem to be quite a bit more uniform than the huge variation that we see in response to natural infection. That's to be expected if that variation is generated by sort of the dose of antigen, because of course with a vaccine everyone gets more or less the same amount of antigen. The durability, it's a question that's just going to have to be addressed empirically. These RNA vaccines, they're sort of new, at least to humans, and we will watch with interest how durable those protective responses are. I would say, however, that if it does end up that we have to go for a booster every year, that at this point we'd still regard that as a win. I think the news that we have is pretty much as good as it possibly could have been, based on where the stage of those vaccine trials are. I'm very, very optimistic. The durability, we'll just have to wait and see. It might be that other vaccines down the track do even better, but we'll see.
Indeed, let's wait and watch. So the next question is, let's see. Okay, it's about the elite responders. So do the elite responders have anything in common? I mean, I know you talked about male versus female difference in the severity with infection, but what about age? And adding to that is, now there are studies that show a relationship between the levels of interferons and the severity of COVID. So did you see any relationship between the interferon levels and the levels of neutralizing antibody?
Okay, so that we haven't measured yet, levels of interferon. The sort of various demographic characteristics, you're right, males, females, severe versus mild disease. Age is a little bit complicated one because what makes someone more susceptible to disease in terms of age is probably a reflection of their poorer ability to mount an immune response. So there are in some studies some correlation of age with antibody response, but not as strong as you might expect if that was simply a reflection of severity of disease. So obviously, if your immune system gets on top early and suppresses the virus effectively without actually mounting an enormous immune response, then you're going to have what looks like a kind of weak immune response, but do well. Conversely, if you get if your immune system is not very good early on and the virus gets on top and gives you a very severe case, then you're sort of, in a sense, smoothing out that what I would anticipate as a strict correlation between the strength of the immune response and how well you would do, because there are sort of factors working in opposite directions, if you see what I mean.
Hey, Paul, I wanted to follow up on that question. So you did find a big gradation of response in the overall neutralizing titer. That's the, what Serrato was talking about, the elite responders. My question is about the 25 patients that you picked for follow-up characterization for escape mutations. Did anything distinguish those people?
Okay, so we picked those 25 for purely practical reasons. It's just easier to select a resistant virus if you've got a more potent selector, if you see what I mean. Basically you're trying to sieve out resistant mutants, and if your sieve isn't very effective, then that's going to be challenging. So there's nothing special about them other than they had good titers of neutralizing antibodies. But even within that group, you find some that look like they have monoclonal antibodies in their plasma, and you have others that look, you just can't generate resistance. So at this point, I don't really know what underlies that sort of variability. And of course, it's going to be quite complicated, the amount of antigen, and then you've got human genetics on top of that, which I don't think we've really begun to explore.
Yeah. Okay. So I've got a million more questions. Want to remind the audience, if you have a question, either raise your hand or type it into the Q&A box. Serrato and I are happy to dominate the Q&A, but please chime in. Paul, going back to the common cold coronaviruses, I was really struck by the observation that the OC43 serum, which is a beta coronavirus, was able to take out the two alpha coronaviruses without, with only like a, half a log worse response than OC43 itself. Like, how in the world is that possible?
So I think that's kind of misleading. I didn't show all the data in the interests of time, okay. But OC43 is very common, but the other viruses are also very common. So we think there's multiple different responses in those individuals. It's just SARS-CoV-2 isn't one of them. Okay.
Okay. So you think even though it was labeled as OC43 serum, it was probably all four common cold coronavirus serum?
Well, yes. I mean,
okay.
Those were simply the PCR diagnoses that were made a few weeks or months before that plasma was taken. But these viruses are so common that I mean, you're probably seropositive to all three. Me too, right? Or at least there's a good chance that you've had all three.
Yeah. Okay, Sarada's got another one now.
Yes. So here's a question to you about reinfection. So do you predict a relationship between reinfection, the probability of reinfection, and the levels of neutralizing antibody?
Yes. So what we already know about the seasonal coronaviruses is that correlation exists. Okay, as the antibodies wane, the probability of reinfection increases. There are, at this point, anecdotal reports of reinfection in SARS-CoV-2, but where it's been examined, where the samples have been available, the reinfection have been in people who've had little or no antibodies at the time, or at a pre-second infection sample. There's also this sort of quite famous case of the boat where a whole bunch of people got infected except one person who had, or one or two people who had previously had infection and had neutralizing antibodies on board. So whether it's actually neutralization of virion particles that is the protective response, or whether it's other functions of antibodies, ADCC, for example, that also contribute, or whether they're simply correlates of some other immune response that's protective, or all of the above, we don't really know yet. What we do know is that neutralizing antibodies are a good, at least in the seasonal coronaviruses, they're a very good correlate of protection, and very likely, but not, I would say, definitively proven mediator of protection for infection. What we can also show, of course, is that you give animals antibodies, neutralizing antibodies, they're protected, right? Some data I didn't show is that protection is more robust if the Fc function of the antibodies is maintained. You mutate away Fc gamma interactions that recruit effector cells, the potency of some antibodies is decreased.
Interesting. So Paul, of course, we have many, many more questions, but I just want to make sure that you're okay with time.
I am okay with time. I don't have anything till 2 o'clock.
Fantastic. Lauren?
I will finish a bit before that.
Okay, yeah. Can we dig more into the gut response? Is that is fascinating and somewhat terrifying, and I guess not all that surprising in retrospect. I mean, certainly the gut biopsies, that's going to be a super low throughput way to characterize patients. But do you think that most people that are exhibiting this ongoing B cell maturation, do you think that they all have persistent populations in the gut or some other reservoir?
It's just too early to say at this point. This continuing maturation of the antibody response does strongly suggest persistent antigen, at least. That doesn't necessarily mean an ongoing infection. We know that follicular dendritic cells can trap antigen, and the antigen can persist, at least theoretically, in the absence of viral replication. So what contribution that apparently ongoing replication in the gut makes to the antibody maturation, we're just going to have to wait and see. It is something that Saurabh and Michelle and we will be studying in the future. But as you correctly point out, it is quite a difficult place to sample systematically and repeatedly. It's not that much fun, to be honest, to give a gut biopsy. But to the extent that it's possible, we will be doing that. Of course, it's also a huge anatomical site, and it's absolutely possible to miss the spots of virus replication when you're taking a biopsy. So difficult to be definitive, but it sort of makes sense that prolonged replication there would help.
I was going to suggest something, super low tech, like just grab a stool qPCR, every time they come in, and if the stool positive or negative correlated with the B cell maturation, I mean, that you're right. I mean, it doesn't prove it, but that would be highly suggestive.
Yeah. So I have to say that is something that one would need to look at are there enough sites of replication in the gut to give a sporadic or consistent signal in a stool PCR. We know during the conventional window of infection that stool can be positive and really quite strongly positive. These sort of later time points that's you're right, a very if it works would be a very good low-tech way of trying to monitor what's going on. There's always this sort of uncertainty whether you find something by PCR, does that really reflect viral replication? I mean, even if you see an immunofluorescence picture of a cell with viral antigen in, is that really an infected cell or a cell that's just eaten some protein? My suspicion is it's real virus replication, but
I mean, there at least have been a handful of instances of isolation of infectious virions from stool, but I mean, it was a somewhat artificial setting.
Yeah, stool's not the most hospitable place for virus. The mucosal lining of the gut, probably much better for it.
Yeah. Well, what are your thoughts on mucosal immunization? And I know Akiko Iwasaki has been saying, since the beginning that, she was hoping that someone would develop a nasal vaccine that we could all get in addition to this. I mean, do you think we need like gut vaccinations as well?
I don't think we do. So there is one paper saying that if you gave the vaccine mucosally, you got a better response or more protective response than if you gave the vaccine in the conventional way. And it's also true that, of course, where the virus is replicating, the mucosa predominantly, that IgA is the more prevalent immunoglobulin than IgG. But it's not true that there's no IgG in mucosal surfaces, right? And we know that serum antibodies probably aren't really doing anything in the serum. They actually have to get to where the virus is replicating to work. They are a good correlate of protection. I am not, sufficiently knowledgeable, I think, as an immunologist. I'm a virologist, Antibodies and virus particles together I'm getting close to my limits in terms of knowledge of immunology. Whether that is a necessary step to generate long-term protective immunity that's a better question for an immunologist. We do know that the antibodies themselves, the antibodies that have class switched to IgG, they've often class switched to IgA as well. So you're in a sense measuring strongly overlapping specificities. So even if the serum antibodies aren't the antibodies that are doing the job, they are in a sense reflective of what's going on at mucosal surfaces. So honestly, at this point, I think we need to sort of get something out there that works. If it works for 6 months or a year, fine, and then we can worry about the finer points of immunology later.
Okay, so I had one more, and then I'm going to try to squeeze one more in, and then we'll let you go.
Yes, back to antibody response. So studies have shown that disease severity is linked to individual genetic variation. So my question to you is, how much are variations between individual, the genetic makeup, how does that correlate with their antibody response? In other words, are some individuals just genetically predisposed to be better neutralizing antibody producers?
That's not clear yet. And we certainly haven't looked at it in our cohort. It's almost inescapable that there will be some genetic influence on antibody production and generation, but to be frank, that what those genes are in terms of the response to SARS-CoV-2 has not been answered yet, at least not to my knowledge.
Okay, thank you. Lauren, do you have one more?
Yeah, so you told us about the boat. Can you tell us about the minks now?
Yes.
You know, both in terms of general concern of them being a reservoir and maybe any insight as to why they are the only species known so far to both get it and to transmit it back to humans. And then maybe also to speak about mutations and how worried we should be about that.
Right. So why mink? I don't know. That is purely a chance thing. The fact that this virus was transmissible to humans from bats, that's just the luck of the draw in terms of what evolution has endowed us with in terms of ACE2 sequence, which seems to be a pretty important determinant of host range. So the mink, what we need to recognize for the minks is the way that they're farmed. They're present in huge numbers in these huge sheds, sort of arranged in sort of the same way as you would do an experiment in the lab with ferrets to see if you could get airborne transmission, right? So lots of minks living in close proximity. There is, you know, a number of carnivores, right, seem to be susceptible to SARS-CoV-2 infection. So domestic cats, for example. I may be wrong. I just can't think of another carnivore species that would be kept together in such large numbers as minks. There probably is one, but I just don't know it. So the fact that minks are susceptible, that's just a roll of the dice, and maybe a reflection of carnivores in general. Now, what does that mean in terms of should we worry about it? I mean, any reservoir species, right, like that isn't a good thing. You know, you're giving the virus an opportunity to explore genetic diversity. I mean, there's so many coronaviruses in nature that have jumped to humans. I mean, we know of the seasonal coronaviruses. What might happen if we let this virus just replicate in an uncontrolled manner in any animal species? You're just doing an experiment whose outcome is unknown, basically. So it does appear, based on the appearance of particular mutational signatures in humans and in mink, that obviously humans transmitted to mink, and it may have come back from mink to humans with a change, right? That change, I think, has also occurred independently in humans. It's in a part of the RBD that such that it could have arisen to better allow the virus to better replicate in mink, and what that effect would have on retransmission to humans isn't clear in terms of ability to use ACE2. It could also be that is a mutation that was selected by antibodies. It's in the same vicinity of the changes that I showed you cause resistance to some antibodies. It isn't exactly the same as any of the changes I showed, but it's clearly in that vicinity. But as I also think I showed, the likelihood of a single amino acid change causing broad resistance to antibodies that are present in the human population generated by natural infections or by vaccines is low. Okay, I don't think we should worry about a mink virus, being resistant to all the vaccines that are being generated. There might be some change in sensitivity, but it's, I would say, extremely unlikely that it's going to make the vaccines ineffective.
Yeah, well, that's a hopeful note to leave it on. Thank you so much, Paul. That was really amazing.
Thank you, Paul.
Thank you very much for listening. Thank you for the invitation.
Thank you.
All right. Take care. Stay safe.
You too. Bye-bye.